And today I will be talking to you about my research for my dissertation, which is testing the potential of character displacement to drive the evolution of root traits. And so my goals today is not only to talk to you about this, but to also, it's kind of dark, is there any technical problems? Okay, anyway, as I was saying, my goals today are to talk to you not only about my dissertation, but to hopefully convince you that this is an interesting study that should be examined and lacking in literature. And so quickly, character displacement is a process wherein species evolve to become more different as a response to intense competition for living resources. And so it can have important implications such as lead to the evolution of novel traits, enable species coexistence, even trigger adaptive radiation, and can potentially lead to speciation. The traditional case is demonstrated on the right by a clip art image of the Galapagos finches, Darwin's finches, where these birds evolved in beak morphology and size as a response to intense competition for seeds. Despite the importance of character displacement, there's just a handful of studies in plant system literature, and most of these focus on above-ground interactions and traits, specifically floral traits. For example, the evolution of floral morphology and color as a response to competition for pollinators. Yet despite the importance root traits play in mediating below-ground competition and resource uptake, to my knowledge, there is no research explicitly examining this question: does character displacement drive the evolution of root traits? And so my main question is, can character displacement drive the evolution of root traits? And I'm addressing this question by testing for the criteria. So I'm trying to see if there's overlapping phenotypic variation between species, genetic variation underlying phenotypic variation, and these two are critical for evolution by natural selection, but what is critical for character displacement is the demonstration that selection actually favors individuals that are more dissimilar in their resource traits relative to those that are more similar, have higher fitness. So essentially, as demonstrated in this prediction, that individuals with greater phenotypic distance have higher fitness. I'm examining this question in a morning glory study system of Ipomea heracia and Ipomea purpurea. And these are ideal because these species are closely related, they're sister species, and they commonly co-occur in the eastern USA. And they've been known to co-occur for about at least 100 years. They have short growth seasons, 3 to 4 months between germination and flowering. They're morphologically diverse and share many natural history traits. So in 2018, I did a field experiment where I grew replicate maternal lines of Ipomea purpurea. This was my focal species, in the presence and absence of interspecific competition with I. heracia. Specifically, in competition, I examined every possible pairwise maternal line by species combination for 10 maternal lines of Ipomea purpurea, 6 maternal lines of Ipomea heracia. So this is 60 unique combination pairings. I had a total of about 1,120 plants. And as you can see on the image to the right, I grew these plants in competition, growing up stakes, where the stems at the soil surface were about 4 inches apart, so they would compete below ground. However, I grew them in such a way that they would not compete above ground. So half of these plants were assigned to be excavated for root phenotype, which is a destructive method, and the other half was maintained to collect fitness data as seed output. For root phenotyping, I adopted the Chauvelomics technique from the plant breeding literature, which is essentially taking soil cores from these individuals, cleaning their roots, and doing some kind of standardized imaging procedures, such as using a camera stand and an imaging box. And this allows for the automation of root quantification. I used a program called DIRT, which is an excellent program that is open source online. It can be used across various computer platforms, and it is continually maintained. This program gave me 78 root traits in total. However, many of these were not applicable to my system because they're for either monocot species or for excised roots, and I used whole root systems. In addition, there was a lot of redundancy in my traits, so I filtered to a total of 33 traits to work with, and to reduce the dimensionality of these 33 roots, I used principal component analyses. and I focus on the main principal components as my root system variables. As you can see from the scree plot, the first 4 principal components explain at least 10% of the total variation, so I focus in on these. For the first principal component, I see that D% traits loaded most strongly. And so these D% traits essentially explain the root system width accumulative to the percentage of soil depth. So this is telling me something about the silhouette globally about the root system, which I refer to as root topology. And the second principal component was loaded most strongly by stem diameter, max tissue angle, vertical root distribution, max width, basal root angle, and adventitious root angle. This is telling me something about the spatial structure of the root system. So I term this architecture. The third component area, so I term this size. And the fourth component are root tip diameter, tap root diameter, and root number. And because this is describing the root system at an individual level, I term this as root morphology. And these are terms that are broadly defined in the plant breeding literature as well for root traits. When we examine the 3D dimension of these 3 PCs, let me just mention quickly that for root area, I'm excluding in the subsequent analysis because previous research has demonstrated that root size is not selected for in below-ground competition between these species, and I have not found maternal line variation for this trait of size. So I'm focusing on topology, PC1, architecture, PC2, and morphology, PC3, and this is what's shown in the 3D scatter plot. And what I want to show here quickly is that these individuals between species vary considerably, and they also overlap, indicating that these species have conserved niche space below ground. Linear mixed model ANOVA reveals that root topology and architecture vary significantly by species, but however we see overlap, indicating that these species can still compete for resources below ground. Further, I found significant maternal line variation for root architecture and morphology, which is the second criteria I mentioned and critical because it suggests that there's genetic basis for these root traits and that they can potentially respond to selection. And so for this critical question and prediction, is there a positive relationship between how different 2 individuals are and their fitness? I examined this relationship. But before I go into the selection analysis, I want to mention that I found a significant effective competition where 16% seed reduction was found in individuals that were in competition relative to individuals that were grown alone. And briefly, how did I calculate phenotypic distance? So I used the first dimensional, second dimensional, and third dimensional distances as the Euclidean distance between within PCs, for example, PC1 minus PC1, PC2 minus PC2, etc. And then every possible pairwise Euclidean distances for PC1, PC2, and PC4, which is topology, architecture, and morphology. However, I did not find evidence of a linear effect between 2-dimensional and 3-dimensional traits onto relative fitness, so I focused on the first dimensional phenotypic distance. And so what we're expecting to find, this is very important, so I reiterate, is that individuals that are competing with those that are more similar, as demonstrated here as this blue point and root picture, will be performed less than individuals that are more dissimilar in their root structures, as shown by the yellow dot. So for root topology and root morphology, phenotypic distances, I did not find a linear effect with relative fitness. However, for root architecture, I did find a significant positive effect between phenotypic distance in this trait and relative fitness within Hymenoptercaria. And so what I want you to kind of stop here for a moment to really focus on this, because this is really important for character displacement and the prediction that selection favors individuals that are more different. Every point here is an average of 8 biological replicates of maternal line by species combination, where those individuals that, as you can see on the left, are more similar in their root architectures than those individuals on the right in the x-axis. And so this meets that criteria that there is a positive relationship between phenotypic distance and fitness. A little bit about root architecture. So as I mentioned a little bit earlier, root architecture is describing the spatial arrangement of the root system. And this is very important because the soil environment, as some of you may know, is a very very heterogeneous environment. For example, immobile resources such as phosphorus accumulate at the topsoil, and mobile resources such as nitrogen accumulate in deeper soil levels. So depending on your root structure will influence how much you overlap with another individual in their resources. And ecology and ecosystem studies in plant systems have demonstrated that variation in rooting depth contributes to plant productivity and community structure. And with that, I want to conclude quickly by reiterating some of the results and key points of my findings. That species, the morning glory species here, had overlapping variation, indicating that they can that their niches are conserved. That root traits exhibit mature line variation, so they can potentially respond to selection, which is important for evolution overall, but also a criteria for character displacement. And lastly, that selection favors an increase in phenotypic distance, which indicates that character displacement is a viable agent of selection. Thus, character displacement may potentially be driving the evolution of root traits in these species in natural populations. However, this is the first study that I know of, so we definitely need more research to know how important and widespread this phenomenon is, and is it occurring in natural populations. Lastly, I want to thank all the members involved that have helped me throughout the years, especially the Bauckham Lab, my advisor Gina Bauckham, graduate students, a number of undergrads that helped me through the Doris Duke Scholars and Europe program, employees at the Mathai Botanical Gardens, and my funding sources. And with that, I'll take any questions, time allowed. Thank you.
Are there any like single traits? You showed your principal components and generally what loaded onset and what you named them. Like from a functional perspective, are there any individual variables you had sort of like functional predictions about how differences among the two species might relate to fitness? And did you look at those like univariate comparisons? I'm not a plant person, so I don't know.
No, that's a really excellent question. So in previous research, I focused on some main traits. I did not do this whole modular approach where I found evidence for selection in root angle, just basically the angle at the top root of the root system in Icomia heteracea when it was in competition with Icomia purpurea, indicating that potentially these species are competing for above ground resources. I am in the process of examining these traits individually to see what may be functionally responsible for these trends. But to me, working with such high dimensionality is something new, so I'm trying to figure out what techniques to use to kind of parse out what traits are responsible, right? Because 33 traits is a lot, and you can't really run so many models without, losing power. So, any questions?
So those are two species of morning glory, and their distributions overlap, or are they widely separated in their natural populations?
In natural populations, excellent question. So, I've done a common garden greenhouse on various populations in these species, and what has shown is that there is significant species variation, but when you look at things like box plots, histograms, they're largely overlapping. So there are still a lot of individuals that retain similarity.
What I'm getting at is if in natural populations they're in different areas, they likely have different microbiomes in the soil. And so you might want to look at microbial improvement.
Well, I ran out of time, but I'm definitely looking into that, but that's something I didn't discuss here, but one of my thesis chapters.